Good day and welcome to Letters and Politics. I'm Mitch Jeserich. Today we are going to be in conversation as how humans have affected the planet really since the species began. My guest for this conversation is Beth Shapiro. Beth Shapiro is a professor of evolutionary biology at the University of California, Santa Cruz. Her work centers on the analysis of ancient DNA. We'll talk about that. She's the author of the book, How to Clone a Mammoth, and her latest, Life as We Made It, how 50,000 years of human innovation refined and redefined nature. She joins me over Zoom. Beth Shapiro, it is a very good pleasure to welcome you to this radio program.
Thanks for inviting me.
How 50,000 years of human innovation refined and redefined nature. I want to begin by asking what's significant about the 50,000 year mark. Is it that something spectacular happened 50,000 years ago, or does it just get harder to tell the story past that period of time? Because I guess the species, you'll correct me, is about 300,000 years old or so.
Yeah, it's actually was an argument between myself and my publisher. What number should we use that actually makes sense, is easy to say, that we really feel like we can go back behind? And so we settled on 50,000 years. Really, we've been impacting species for longer than that. And in my book, I do talk about before 50,000 years ago. And I start really with the origin of our species and in Africa, and how there were lots of different human-like species early on. And then we move out of Africa sometime, well, possibly twice or possibly several times. The fossil record keeps getting more and more detailed as remains are discovered in places around the world. But we know that there was a mass exodus somewhere around before 60,000 years ago that really led to the distribution of people as we know them today, kind of the beginning of that. But people did, it seems, leave Africa before that. I think another reason to pick 50,000 is this is really when we start to see in the fossil record the very first impact of these large numbers of people that moved out of Africa and then into Eurasia and then eventually populating the rest of the world. And that impact is in killing off other species and changing habitats and landscapes as we encounter them. We know that as people moved into Eurasia and then North America and later onto islands, that the appearance of people in most of these places corresponds to a period of mass extinctions, mainly of large animals. And this is what we observe in the fossil record. So I guess that's why I pick around 50,000. It's not perfect, but you gotta pick a number somewhere, right?
Is my understanding also correct that it gets more difficult to measure fossils that are older than 50,000 years?
Well, what it gets difficult to measure is how old those fossils are. We can find fossils, and in some environments, fossils are well preserved that are much older than that, even with DNA in them. And we can talk about that in a bit. But the procedure that we use to figure out how old a fossil is called radiocarbon dating for the most part. And this takes advantage of the fact that as carbon hits the Earth's atmosphere, we actually, the plants, animals that are growing uptake two different forms of that carbon. And one of them is radioactive, and it decays to the non-radioactive carbon with a predictable half-life, and we can measure the exact amount of radioactive and non-radioactive or stable isotope of carbon in these fossil remains, and that can tell us how long ago that animal was taking up carbon in the atmosphere. So that's what we use, and we really lose power with that approach around 50,000 years ago or so, and that's just because there's just not enough of it left to measure with any accuracy.
So that's how we do it. We measure decay of the carbon.
That's right, the radioactive decay of carbon. Yeah, I mean, there are other ways to figure out how old a fossil is. Sometimes, I work a lot in the North American Arctic, and we sometimes find bones that are underneath what looks like a little thin layer of a little thin white blanket that's in this otherwise dark mud profile. And we know that if that little thin white layer is undisturbed and the bone is beneath it, that this is a layer that accumulated over time with the oldest stuff on the bottom and the youngest stuff on the top. And we could use different approaches to guess how old that how old that little white layer of blanket is. And that is normally a volcanic ash layer. And so when a volcano erupts, it spews all these fragments and out into the air, including ash. It settles down into a big blanket, and then that gets covered up with dirt later. So we can know using other methods how old that blanket is when the volcanic eruption took place, and any bone that's underneath it is older than that. So we've worked with bones in the Arctic that are around 700,000 years old, and we know that because they're underneath one of those blankets, an ash layer from an eruption that happened around 700,000 years ago.
Underneath the soil, it's a big timeline.
That's right. Yeah. And if you get lucky, it's undisturbed. Now, in a lot of places, it is disturbed. The soil moves around, it freezes and thaws, it cracks, and water gets in there and washes stuff around. So we can't always know how old something is just because of where it is in a sediment column. But in some special circumstances, we can. And that's great for us because it gives us a great way of knowing how old something is that's older than 50,000 years or so that we can date with radiocarbon.
You mentioned a mass extinction about 50,000 years ago. What can you tell me about that?
Well, that's the beginning of an extinction event. We see, for example, when first people get into Australia, which is just before 60,000 years ago, we start seeing in the fossil record evidence of the large Australian megafauna that don't exist anymore, becoming extinct. And then we see in Eurasia, when people are first there, evidence in the fossil record that large animals like woolly rhinos are becoming extinct and other types of rhinos. There's long been a debate about the relative culpability of people compared to climate change in the extinction of these megafauna, because it so happens that in a lot of places, in the New World, for example, and also in Eurasia, when people first turn up is also a period of very tumultuous climate change. And so it's hard to say with certainty whether people turned up and hunted everything to death or changed the habitat so much that the species couldn't find enough food or enough space to live, and it was our fault that they went extinct, or because the climate was, for example, in North America, rapidly warming out of the peak of the last ice age, which may have been what caused the animals to go extinct. I think there's probably some truth in both. Clearly, the climate was changing, and this was going to be impacting animals. But the Pleistocene ice ages were a period of about at least 20 different oscillations between really cold ice ages and really warm interglacials. And only one of these climate transitions was associated with a mass extinction event. And that just happens to be the one that also included us turning up, or our ancestors turning up, and being that maybe proverbial straw that breaks the mammoth's back.
The earth warming made it easier for us, I guess, to leave.
Well, in some ways, yeah, but not all human movements were tied to warming events. When our ancestors were moving into Eurasia, this was an interstadial period. It wasn't a really warm period. It wasn't the peak of an ice age, but it was a cold period, a climatically tumultuous period. And it could be that some of that unpredictability of climate was one of the things that was driving people out of Africa and to look for new habitats to settle, because maybe they were running out of space, or populations were changing, or they were running out of whatever it was that they were eating at the time. But it wasn't just warming events that made it easier. We were explorers from the beginning.
Do we see, and of course, we've had climate change in the last 50 years several times. Do we always see an extinction event that closely relates to that change?
No. Like I said, the Pleistocene ice ages were lots of different shifts between very cold ice ages and very warm interstadials. And we really only see this mass extinction event in this most recent time period. There's also, people were moved into Australia earlier than they did to Eurasia and into North America. And when people moved into Australia, there isn't any evidence of any really rapid climate changes happening at that time point, but there is evidence of extinctions. And much later on islands, people are exploring and looking for islands and moving into new landscapes. And we do see, when people turn up on islands, there's tons of evidence of large animals, like in New Zealand, the moas, or on Mauritius, the dodo, becoming extinct. but there is no simultaneous rapid climate event. And so in these situations, both the oldest in Australia and the more recent on islands around the world, we do see evidence of humans turning up and things going extinct in the absence of any dramatic climate change. So to my mind, these are the most compelling examples of really the impact that we could have on species. And I think what's important to say here is that at the time when people were first moving out of Africa and into different parts of the world, they weren't deliberately causing these extinction events. They weren't going into a place and thinking, oh, I'm going to eat all the moa, or oh, I'm going to eat and kill all the woolly rhinos or giant short-faced bear. Well, they might have been thinking, I'll kill all the giant short-faced bears because these were scary things, right? So maybe they didn't want those in their backyard. But for the most part, these extinctions weren't deliberate, right? We didn't know. Our ancestors didn't know they were causing them. That's one of the things that separates what's happening today from what happened in the beginning. Now we see that we're driving species to extinction, and we also know that this is our fault, and we have a choice to make in what we're going to do. But we can get toward the future later on.
And we will, because that's interesting too. But my previous question was, does climate change, do we see a mass extinction event come with it each time? I guess my second question to that, then, do we see whenever humans show up anywhere, a mass extinction event occur, or are there exceptions?
I think we see extinctions whenever people turn up for the first time in places. There are exceptions in that there are some species that have survived humans turning up, even some megafaunal species that might be surprising. But to answer your question, does the initial human appearance on a place coincide with records of extinctions in the fossil record? The answer is yes, it does.
That's interesting, and probably not surprising if you really think about it, but still is telling. Now your work centers around ancient DNA. This is somewhat of a new field that is really sort of revolutionized and changed our understanding, and even upended some of the previous things that we thought we knew, especially from sort of regular archaeology. Tell me more about what the study of ancient DNA means.
Well, it's a relatively new field. I guess the very first ancient DNA paper that was published was in 1984, so not super new, but still pretty new. Scientists working at Alan Wilson's lab at UC Berkeley managed to get a small fragment of DNA, isolate a small fragment of DNA from the preserved skin of an animal called a quagga. And this is an animal that went extinct relatively recently that was living in South Africa. And it's a relative of a zebra. There are pictures of them and they look like zebras. So it wasn't actually that surprising that they could show using this DNA that the quagga was a type of zebra. But this was the first time that anyone had recovered DNA from an animal or plant that wasn't still alive. And the idea that DNA can survive and persist post-mortem sent shockwaves through the scientific community. And there was a race on to try to figure out what the oldest and coolest or most interesting or most spectacular or even crazy specimen that people could get DNA from would be. And over the next couple of years, or decade or so, there were people who published DNA from dinosaur bones, from insects entombed in amber, from really old, like hundreds of millions of years old fossils of leaves and things like this. And all of those very, very old DNA studies have something in common, and that is that none of them were real. It turns out that after an organism dies, the DNA that's preserved in their cells and the cells themselves start to break down into tinier and tinier and tinier fragments until there's nothing left or nothing that can be recovered anyway. And in most instances, this happens relatively quickly over months or even years. But in certain environments, like really cold places or places where the temperature is stable and there isn't any light, DNA is broken down by solar radiation, by freezing and thawing, and by microbes that get into stuff and chew it up, just eat up all the materials and DNA. But if the sample is somehow protected from that, then DNA can survive for a while. The oldest DNA that has been recovered to date, we published with colleagues from the Swedish Natural History Museum, a study led by Love Dahlen. And this was Mammoth DNA from a tooth that dates to around 1.2 million years ago. And this broke all of the records. As I say, normally in
Much
the
longer than that
Arctic.
50,000, much longer than that 50,000 number we were talking about. Yeah.
Right. Yeah. Well, yes. Well, and we knew how old this mammoth tooth was because of other context, including things like the volcanic ash layers, like I was telling you about, because we certainly couldn't have gotten a radiocarbon date on a mammoth tooth that's that old. But we were pretty surprised that DNA remained in it. We know that there are rare instances where we can get DNA from things that are old. We've been working with some horse remains from the Yukon that we know are around 700 to 750,000 years old. And we have gotten whole genomes, like short fragments of DNA, but billions of these short fragments of DNA that we can piece together and make a whole genome out of from a couple of different horses from that time period. But those are exceptional. So this mammoth was particularly exceptional. Most of the things that we work with are within 50,000 years. And they're also from cold habitats, working with things from the lower 48 United States where it wasn't frozen through most of the ice age. We tend not to get stuff that's very old at all, maybe a couple thousand years in exceptional circumstances like caves, maybe 25, 30,000 years, but mostly not really. So all the dinosaur stuff, dinosaurs, non-avian dinosaurs anyway, went extinct more than 65 million years ago. So we're not going to get any dinosaur DNA out of any of these remains.
Does DNA then must, correct me, I'm just guessing here, must decay then more slower than, say, the rest of the human body other than bones? Or maybe you find DNA in bones?
We find DNA in bones. So yeah, the bone matrix breaks down more slowly than the other parts of the bone. So the bone will break down more slowly than tissues. But the DNA that's in the cells of the bones, that's where we're finding it. We actually take a piece of that bone and we ground it up into a fine powder. And then we use enzymes to lyse those cells, to open them up, to try to release the DNA. And the DNA within them is broken. It's, if I were to take a swab from the inside of my cheek, I could extract DNA fragments that are millions, maybe hundreds of millions of letters long, just one big molecule. But with ancient DNA, we're lucky if our fragments are maybe 30 letters long. So we're working with something that is way terrible compared to modern DNA.
And you have to reconstruct it.
That's right. No, so I liken this to you're putting together a puzzle. Let's say you want to put the mammoth genome together, and somebody's given you a puzzle, and it's about a 100 trillion piece puzzle, right? Or actually, let's do it this way. The mammoth genome, we know, is about 4 billion letters long, right? And all of our fragments are maybe 30 billion letters 30 letters long, but they overlap. So it's just a bunch of cells that have copies of the genome. They're all broken up into smaller fragments. So we have all of these puzzle pieces, 30 letter long puzzle pieces that we're trying to use to piece together a 4 billion letter puzzle. But when we extract DNA from a mammoth bone, maybe if it's a good bone, a third of the DNA that we get is mammoth DNA, and the rest of it is stuff from bacteria that live in the soil, or plants that might have grown into it, or fungi that might have grown into it while it was going. So you got this puzzle, this box of puzzle pieces, and there's a whole bunch of mammoth DNA, but that's about a third of the puzzle pieces that you've got, right? And you've also got some fungi, and you've got some other things. You've got more puzzle pieces than you need. And the picture on the front of the puzzle is of an elephant genome, right? So it's not even an exact match of the thing you're trying to piece together, right? Because we don't know what a mammoth genome looked like, but we do know what an elephant genome looked like, and we can use that as a scaffold to try to figure out where our 30-letter pieces go. It's a mess, a hot mess, right? But using computers and fancy algorithms, we can do it. And in fact, we've pieced together a whole bunch of whole genome sequences from species that used to be alive, including mammoths, but also Neanderthals, which have taught us a lot about what it means to be human, and things like ancient bison and ancient bears and woolly rhinoceroses. And we're using these things to really learn, how it was that people and climate change impacted species and ecosystems that used to be alive.
So we have to compare it when we're reconstructing it to the closest living animal today.
Yes. Yeah.
This is Letters and Politics, and we are in conversation with Beth Shapiro. Beth Shapiro is a professor of evolutionary biology at the University of California, Santa Cruz. Her work does center around the analysis of ancient DNA. She is the author of the book, How to Clone a Mammoth. Her latest is called Life as We Made It, how 50,000 years of human innovation refined and redefined nature. I did want to ask you more about the Neanderthals because this is pretty fascinating. It's also pretty fascinating to imagine living during a time in which you weren't the Homo sapien was not the sole human species that existed. There used to be others that lived all around the same time. We didn't evolve from one another. I guess we outcompeted the others.
Yeah, we were evolutionary cousins with the Neanderthals and Denisovans. We share a common ancestor, but that common ancestor is no longer. And Neanderthals moved out of Africa before the common ancestor of anatomically modern humans. And Neanderthal populations were already established across Eurasia when people, anatomically modern humans, people, that's what we like to call the lineage that led to us, left Africa. And we didn't know, although there was some suspicion for some time looking at some of the fossils, what happened when people first left Africa and encountered Neanderthals. We're pretty sure that one of the consequences of this meeting is that Neanderthals no longer exist, right? So we outcompeted them, we drove them to extinction in some way. But what we discovered when we first were able to isolate DNA directly from Neanderthal remains is that we also had a different interaction with them, and that all of us who are alive today have some ancestry that dates back to admixture, hybridization between Neanderthals and older human lineages, and the early modern humans that were both in Africa and after leaving Africa.
So what does that mean?
It means that when anatomically modern humans left Africa and encountered a Neanderthal population, sometimes they got together and things happened. And we don't know. I mean, would it have been that they decided that they wanted to interbreed, or was it something that was more forced? We really have no idea. We can't go back and figure that out. But we do know that these interactions happened and matings happened multiple times. And we know this because we can look at living people today. And now that we have the sequence of the genome of Neanderthals and of another human that we call the Denisovans, that was first discovered in a cave in the Altai Mountains called Denisova Cave, which is why they're called Denisovans, that there were interbreeding events between anatomically modern humans at several different places in the world as people dispersed into these habitats. And sometimes this was actually good for people. For example, one of the first things that we learned is that there are some populations of people that have MHC alleles. These are types of DNA that make us more fit in the case of being exposed to pathogens. So it's the part of our DNA that helps us to fight off pathogens. We know that there are some parts of the world where populations of people alive today have MHC alleles that came into that population because of breeding with Neanderthals. This makes good sense. If you're going to move into a place where there's already people and diseases, if there's DNA that makes you better able to survive in the face of those diseases, then having that DNA for yourself or for your children will be beneficial, and those alleles will be passed on to the next generations.
You write in your book that we all have Neanderthal DNA in us, but we all have different parts of Neanderthal DNA. Yeah. We all don't share the same Neanderthal DNA.
Yeah, so this is cool. So we know that all of us have somewhere between 1% and 4%, maybe a tiny little bit more of Neanderthal DNA. In fact, if you go and get your DNA tested by any of these companies that'll do that for you, you can often get a ranking. So this is how much Neanderthal DNA you have. I know in my family, there's a good competition to see who has the most Neanderthal DNA, and it's my sister. She has the most. But what we don't know, what tends to be more surprising to people, is that we don't all have the same, 2 to 5% Neanderthal DNA. We all have different bits of the Neanderthal genome that got passed on. And in fact, if you sum up all of the Neanderthal DNA that's present in people all around the world, we can piece together about 93% of the Neanderthal genome. And this is fascinating, not because of that 93%, but because of the other 7% that nobody has. And a big open science question is, what is going on with that 7%? Because in that 7%, some of that's going to be lost by chance. There was some mating that happened in the beginning, and then a lot of it was lost. But there's bound to be all those important things that if a baby inherited the Neanderthal version of that DNA, they just couldn't survive as a human. And so that didn't survive in our population. And so if we could really understand what genes, what things in the genome are in that 7%, we can really start to get a much better understanding of what it is to be human, what is unique about our lineage compared to Neanderthals.
Is it just a matter of time until we figure out that 7%? Is there some breakthrough that we can anticipate will happen?
Yeah, I mean, we're still learning how to decode the genomes of all species, including humans. And what we can do is we can look at that 7% and we can ask what part of that 7% is genes. We know that not much of our genome is made up of genes, which are the things that make the proteins that make us look and act the way we do. A lot of it is just other stuff that's in there, some filler, some spatial organization, some repeats, etc. We don't really fully understand what those all do. But if you start by looking at the genes, you can say, okay, so what is important? What proteins are made out of this part of the DNA? And people have done that. There are lists of the genes that are in that part of the genome, and a little bit at a time, labs around the world are starting to dig into it and figure out what those genes do. So I'd say we're still pretty far away, but we have some direction. We have some exciting new science ahead of us in the next couple of decades or so.
If we all have Neanderthal DNA in us, and this was from breeding with Neanderthals thousands and thousands of years ago, does that, I guess tens of thousands of years ago, does that mean then that there were Homo sapiens that were different than us now that didn't have Neanderthal DNA? Does that make sense?
Yeah, no, it could. I mean, we know that the people that survived are people that bred with Neanderthals, and surely, the way you think about, the way this process works is everybody who's alive today has one single common ancestor. We are all related to one person all the way back in the past. And that person was not the only person alive in their generation. Just all the rest of them didn't leave any offspring. I mean, there's recombination in genomes and everything is passed down, but at every place in the genome, we can trace it back to a single common ancestor, a single common version. And that common version was not the only thing that was alive, and everything except it is now extinct. That's just the way evolution works. So sure, I'm sure there were people around that did not breed with Neanderthals, and those parts of the genomes didn't
died out.
Down. Well,
died out.
Recombination and passing things around. So it's all very complicated. It's a little bit more complicated than that, but because of yes.
You do ask, I think, a very interesting question in your book, Life as We Made It, and that is, what does extinction actually mean? Considering that we do have DNA, Neanderthal DNA within us, and many animals still exist that had previous species, the DNA of previous species that we see as having gone extinct.
Yeah. Well, it's kind of like, what do you call a species? This is one of those things that I think scientists argue about and discuss because it's sort of philosophically interesting to think about. But really, the concept of a species is something that we made up to make it easier for us to do things, to say, oh, well, that is a dog, and that is a cat, and that is a wolf, and that is a polar bear, and that is a brown bear. But that's not really the way evolution works. A polar bear doesn't wake up one day and say to itself, oh, I'm not a brown bear anymore. I'm never going to mate with brown bears again. Instead, normally this isolation, reproductive isolation, happens by chance over some long period of time where they just happen to not interbreed and diverge from a phenotypic and physiological perspective. And then if they come back together again because their habitats shift as climate changes, maybe something has changed that means that they can't reproduce and have fertile offspring. But often that isn't what happened. That's not what happened with us and Neanderthals. We know that's not what happened with brown bears and polar bears. Whenever they encounter each other today or during the last ice age, when their habitats were shifting around, they would interbreed and produce fertile offspring. So they're not reproductively isolated, even though we call them different species, because it's convenient for us to say these two are different species. They look different. They have different behaviors. We should protect them separately. But from a reproductive perspective, that's just one species concept, that probably the one that we're all most familiar with, but not necessarily completely 100% correct. A lot of biology is a bit squishy like that, but that makes it fun.
The bison is your specialty.
It's one of the things I've been working on. It was the second species that I ever worked on as a PhD student. The first was the dodo. I got to extract DNA from the leg of the famous Oxford dodo specimen and use that to figure out that the dodo is a type of pigeon, which was disappointing to many people, but because they wanted it to be something really spectacular. But
Well,
it is spectacular.
If you actually understand a pigeon, it is spectacular. Pigeons are pretty spectacular birds. We just treat them like rats in a city, but their social formation, their ability to navigate is really amazing.
Yeah. But after that, I started working on bison. That also wasn't by chance. And it was the early days of ancient DNA. And everybody had, in the different labs around the world that were able to do ancient DNA, they'd kind of divvied up all the animals by coolness. So Svanzi Pabo, he got Neanderthals. And my boss at the time, Alan Cooper, he was able to work on bears and bison. And there was something else, moas. Well, I don't remember. I don't really remember. But then Ian Barnes, who was a postdoc in the group at the time, he picked bears, and that left bison, which nobody seemed particularly bothered about. And if I'm honest, I wasn't really that excited about bison either. But I started working on them, and they're fascinating animals. They're not entirely reproductively isolated from cattle, so we can use them to figure out what it is that makes bison and cattle unique to each other. They're really fun model organisms for thinking about climate change. So how did species that didn't become extinct in North America during the last ice age, what did happen to them when it got colder and then suddenly warmer again? And when people turned up, and because they're still alive, actually, it's way easier to study them because as a template for mapping their ancient DNA, I can just use a bison, right? I don't have to use an Asian elephant for a mammoth. They're still here. So I can use bison. Yeah.
Yeah. One last praise of the pigeon, only because, I think the fact that a species can flourish in the world today is also something remarkable. And I think we have a way of kind of downplaying any species that is ubiquitous that we see everywhere. You mentioned pigeon and the dodo.
Well, actually, the closest living relative of the dodo is a pigeon called the Nicobar pigeon, which is this gorgeous, super colorful pigeon that's an extremely strong flyer that lives on a bunch of islands across the Indian Ocean. And it diverged from the dodo, and the dodo's closest living relative, the solitaire, some 20 million years ago. So this is an old lineage, the dodo. And presumably what happened was the dodo, or the ancestor of the dodo, arrived on some oceanic island and eventually lost the ability to fly. So when people first discovered the dodo on the island of Mauritius, it didn't have any predators. It was running around on the ground, completely, oblivious to the fact that people were a danger. But it wasn't people, directly that drove the dodo to extinction. It was because people brought with them rats and cats and pigs, and dodos laid a single egg in a nest on the ground. And so these rats and pigs would just eat that egg. And if you can't reproduce, then you're not going to survive, right? So it was within 24 years, they say, of the first appearance of people on Mauritius that the dodos became extinct.
So here we are in the 21st century, and what people are now calling the sixth great extinction. As an evolutionary biologist, as somebody who has done great research on ancient DNA, how does this inform the way you view the state of the natural world today?
Well, I'd like to go back in time a little bit again to answer this question. So I said early on in the interview that when people first appeared in these places that they were naive to, they didn't intentionally drive any species to extinction. It was because they changed the environment. They hunted, yes, in many cases, but they also maybe used fire and they disturbed landscapes, and other species just found themselves confronted with both a new predator and with ecosystem change that they'd never faced before. But that wasn't our ancestors' choice. They didn't go in there and say, I'm going to drive these species to extinction. What happened eventually was that our ancestors, people, discovered that extinction was not necessarily the only possible outcome to trying to keep your family fed and healthy. And we started, once we realized that this was our power, we started to change and reshape our strategies for hunting. Technology had evolved by around 10 to 15,000 years ago, where people were only taking non-reproductive females or only taking males, leaving the reproductive females in these wild populations so that the populations could continue to reproduce and could grow, be sustainable.
Who was doing that? Yeah, who was doing that?
This was, there's evidence of changing of the hunting strategies around the Fertile Crescent by about 12,000 years ago, and other places in the world as well, maybe a little bit later. The earliest was around the Fertile Crescent, around the same time as we start seeing the very first evidence of farming, of people, doing things that mean that crops are going to grow closer to their settlements, of bringing animals closer to those settlements and enclosing them so that they're staying. And then that leads to these technological changes where we start to choose which animals and plants reproduce. We plant the plants that hang on to their seeds longer so that they don't fall to the ground and we can collect them. We choose to breed those animals that don't run away when we come near them, or that are happy to take cues from a person. And this is a real technological change that our ancestors developed at this time point. This was the transition from being hunters to being herders, from being foragers to being farmers. And this is the beginning of our intentionality of really manipulating the evolutionary future of the plants and animals that were around us. And this, the initial driving things to extinction wasn't intentional. Here we see the beginnings of domestication and intentionality. And then much later, but around the time of the turn of the 20th century, we see that there is yet another shift, a technological shift, where people are realizing that these successes as herders and as farmers are really leading to global devastation. The forests have come down. There's hardly any room for our domestic animals, and our population is growing and succeeding. And we begin to see in the political culture of the time that people are very interested in maintaining wild species and wild spaces. And the way that they do this is to make them less wild. So they decide to transition again and use these strategies that they have been using for millennia on our domesticated species, on wild species as well. And this is the beginning of the conservation movement. And, people often think of conservation as, just leaving something alone, letting nature recover, being as natural as possible. But in fact, it's really not that. It's the opposite of that. If we choose to protect, the Florida panther, we do so by importing Texas panthers, counting how many there are, vaccinating them, protecting them from disease. When we choose to protect our national parks,
we'll even kill off invasive species that have now.
That's right. We remove invasive species. We are deciding who reproduces, where they live, how big their populations can be, protecting them from predators and from disease and even from us. We are determining the evolutionary fate of every species that's around us, and not just our domesticated species. And that is what brings us to what's going on to the present day. So now here we are in the face of climate emergency and a biodiversity crisis, and we already are in charge of everything that's there. We are already making the decisions about who's going to reproduce and where everything is going to live. And yet we still have this clash of ideas in conservation, where you have, for example, Ed Wilson and his really brilliant book, Half Earth, where he suggests we just wall off and give half of the earth to the animals and everything will be okay, versus Emma Maris and her ideas about rambunctious gardening, how we have to really capitalize on appreciating the stuff that we have today where it is, and think about the world as we've made it, as we try to protect things. And I fall more on the Emma Maris side here. I think it's a great but naive idea to think that we can wall off half of the earth and leave it alone. Our fingerprints are already everywhere. And
think of the mass migrations and wars and everything that would cause.
Well, yeah, it's impractical from a geopolitical perspective, as well as but it's also impractical from a conservation perspective. I mean, it wouldn't work anyway. So we don't have to even get into the catastrophe that it would cause from a realistic perspective. I mean, it's The pace of habitat change and climate change today far outpaces what evolution can do to keep up. The evolution works at the scale of generations, things that make an organism more fit in the environment in which they are born get passed on to the next generation. If the environment is changing faster than the pace of generations, then it's impossible to imagine that evolution on its own can save these species. Instead, we have to embrace the fact that we have been intentionally changing everything, at least for the last 15,000 years, and possibly longer, if you think about when dogs might have been domesticated, and really recognize that the only way that we can live in a planet that is both biodiverse and filled with humans is if we continue to get our hands dirty.
Now, we've had Emma Maris on this program just a few months ago to talk about her latest book. And again, the idea is support what we have here, including in urban environments, including with gardening and parks and these things. Obviously, by also keeping preserved lands that are out there. But I got the sense in your book that you would go even farther and dealing with DNA itself. Am I correct in that?
Yeah, so I think that, as I've said, this pace of adaptation is something that we can't keep up with, but we do have new tools at our disposal that allow us to actually help species to adapt faster than evolution can do it. And I am talking about gene editing and genetic engineering tools. Imagine, for example, if we could identify what it is that makes some coral species better able to live in warmer waters. Could we then take that bit of that coral genome and put it into other coral species so that multiple species can be able to survive and adapt in this water? There are a couple of ongoing projects. One of my favorite examples is the black-footed ferret. This is a project that's being run by Revive and Restore, which is a conservation-based nonprofit out of Sausalito. They have been working with the San Diego Frozen Zoo and U.S. Fish and Wildlife and a couple of academic partners to see what they can do to help the black-footed ferret not become extinct. So this little ferret, it was one of the very first, one of the original listed endangered species. And in fact, people thought it was extinct. There was a captive breeding program in the 60s and 70s, I think, that failed to make any captively bred black-footed ferrets. And they thought that there weren't anymore. And then in the mid 80s, there was a population that was discovered near Meeteetse, Wyoming. And this population was brought into captive breeding. And with some new information and more updated techniques, they were able to successfully create black-footed ferrets, and they can release them out onto the wild. And this is an amazing, collaboration between indigenous groups and political groups and local stakeholders across the central part of the US today. But there's still a problem. And that is that when you release these black-footed ferrets, they eat a prairie dog, because that's what they do. They eat prairie dogs. And that prairie dog tends to have sylvatic plague, which was imported from Europe and is prevalent among all these different things. And then they die. So you can vaccinate them and release them and then capture them and vaccinate them and capture any native-born offspring. That's not a sustainable conservation program. But there are two approaches that we can use using some of the technologies that are available to us today. And these are the technologies that Revive and Restore have been pushing. First, in the Frozen Zoo in San Diego, there are several different tissue specimens from black-footed ferrets that were from the previous population, so not the population from Meeteetse, so genetically different from them. And about a year ago, Revive and Restore and the Frozen Zoo announced the birth of Elizabeth Ann, who was a clone of an individual who was alive more than 30 years ago. And her DNA is different than any of the individuals that are in the captive breeding program today. She will make, for example, actually an 8th founder lineage, if you can believe that. All living black-footed ferrets will trace their DNA back to 8, including her, finally. And she can introduce this welcome burst of genetic diversity that can help them to avoid some of the impacts of inbreeding. It may not, of course, mean that they can survive the plague. And for that, they're looking to another possible solution. And that is making a transgenic black-footed ferret. The domestic ferret, which is an evolutionary cousin of the black-footed ferret, is naturally immune to plague. It grew up along with it in Europe, and it doesn't get plague if it's exposed. So Revive and Restore, and Fish and Wildlife, and the Smithsonian and some other partners are trying to figure out what it is about the domestic ferret's genome that makes them immune to plague. And when they've discovered this, what they hope to do is use gene editing to transfer that into the black-footed ferret. You'll create a black-footed ferret that is 100% or 99.9999999% black-footed ferret, a tiny little bit domestic ferret, and that tiny little bit will make those black-footed ferrets not die when they eat an infected prairie dog. So this is an example of what I think is a really exciting opportunity to be able to use new tools, our new gene editing and genetic engineering tools to help species to survive and thrive in an increasingly turbulent and human-dominated world.
As you well know, manipulating DNA is scary to a lot of people as well. One, and as you point out in your book, all species go extinct. There's none that won't at some point. But other things is this issue of playing God, but I don't believe in God. But what I do think of when people say playing God is we don't know everything. We never know everything. And to be affecting evolution so quickly, almost turning on a dime, something that usually takes long time to occur and to be successful, it seems to be laden with unforeseen potential consequences.
Absolutely. But there's also an unforeseen or probably foreseen potential consequence of not capitalizing on these technologies. What we have already done is manipulate habitats and climates and species at a pace that evolution can't keep up. And here we have some tools that, if we think about them and allow ourselves to evaluate what risks they bring, might be able to help us save these species from becoming extinct. I completely understand the fear that people have about risks of using these technologies, but I also don't want our fears of these technologies to get in the way of the absolutely incredible advances for conservation and for agriculture that these tools can bring. We've been manipulating things in crazy ways for a long time. I mean, here's an example. In agriculture, people are super scared, and understandably so, that we might insert DNA from one organism into another organism, and that little bit of DNA might interact with the organism's other DNA in some way that we can't predict, right? That's understandable fear if we don't understand what's going on. But at the same time, one of the most common practices in agriculture is called mutation breeding, where you take a whole bunch of plants and you zap them with something that's going to cause them to have lots of mutations. And then you grow them up and you pick the one that you like, and that's what you eat. And there's tens of thousands of mutations that are uncharacterized and unanticipated in those species. Things like Renan wheat and ruby red grapefruit juice. These are all created by mutation breeding. And yet when you go to the store, you see your ruby red grapefruit juice that has that GMO project verified label on it. Yeah, sure, it's not a GMO, but it's a product of mutation breeding, which introduces thousands of mutations, whereas genetic mutation can introduce just one very specific mutation. I don't think mutations are dangerous. That's not why I'm saying this. It's just there's such an inconsistency in the way that we deal with things right now that I think our fear is rooted right now in people telling us to be afraid rather than in genuinely trying to understand what's happening and what's going on. We not only have we been messing with things for a long time, but we've been messing with things in this way for a long time without understanding it. I think there is a tremendous danger in not allowing ourselves to learn more about these technologies so that we can make an educated decision as a global community about what we want to do than there is in there's way more risk in that than there is in giving ourselves the time and chance and space to have these conversations right now before we really can do these things, right? Let's have these conversations and figure out what we can do, what we can't do, what really is scary, what isn't scary, and see what we can do to paint a picture toward a future where we can survive and thrive alongside other species.
We could clearly have an hour-long conversation on this alone, but we are down to our final minute. But I just will ask, because I know people are going to be thinking about this, is that we have a long history of inventing new technologies that at the time seemed to be fine, but give it some time and we end up learning that it had very adverse health effects.
Yeah, I think, domestication. Yeah, sorry. Go ahead. What's your question?
Well, just that's the concern is if we go into changing these things, what is the long-term effect going to be?
Well, I I can't predict, obviously, what the future is going to look like, but there's been a ton of research in actual transgenic agricultural organisms that have shown no adverse health effects. I know that all we hear about are Serralini's rats that get cancer, but that study was actually rejected. It was kicked out. It was retracted because it was shown not to be true. And this is just an example of this really loud voice about how scary and dangerous things are that isn't actually true, right? Let's give ourselves the opportunity to have open minds and actually think about the truth about these things. Don't just listen to the people who are telling you to be scared. I think we live in a climate of that right now. And we can all see, if you look around at, the vaccine hesitancy that's going on, the danger of listening to a bunch of voices that may or may not be telling you the truth. And I think we're also there with gene edited and genetically modified foods. I should say, there's a whole suite of GMO foods or gene edited foods that are out there that don't move DNA between organisms. It just turns a gene off or turns a gene up or move. And so you use these gene editing technologies to tweak the genome, but not to move DNA at all. And that can give us a whole suite of different types of foodstuffs that, non-browning apples, for example, that kind of thing. There's good stuff out there that doesn't involve moving DNA between species, but we don't hear about it because everything that's gene edited or genetically engineered is some crazy thing like tomatoes with fish DNA in them, which don't exist and never have existed. And yet that's a thing that people hear about. Right? It's, keep your mind open. There's some really amazing stuff out there that will be good for us and good for the planet. And sure, there are risks associated with these technologies. They are different than the technologies of our ancestors, but there is a far greater risk in being too scared to even see where these technologies can take us.
Beth Shapiro is a professor of evolutionary biology at the University of California, Santa Cruz, and she is the author of the book that she has joined us to talk about called Life as We Made It: How 50,000 Years of Human Innovation Refined and Redefined Nature. Beth Shapiro, I've enjoyed our conversation very much, and I thank you.
Thank you. It was fun.